Okay, hi everybody. So I'm just going to finish up here. My talk's entitled Simultaneous Cortical and Basal Ganglia ECoG Recording to Test Dissociable Circuits for Conflict and Stopping. So I'm just going to start out by giving a little pitch for something I'm terribly concerned about for 2 minutes, so just bear with me. You all know about this, but not everyone is perhaps acting on it. We're facing a catastrophe, as you know. You can see in this figure here that the temperature and carbon dioxide have gone up and down over 800,000 years in lockstep. But look what's happening to us right now. We're up at 420 parts per million, and the temperature is going to go with it. How fast is the temperature going to go with it? Well, this is the current situation. Where we've had 1 degree of global warming right now, and we're probably going to have 1.5 degrees by 2030. That means a 50% intensification of all storms, floods, fires, hurricanes, and so forth. This is the IPCC RCP 8.5 pathway, which is the worst-case scenario, and that's the scenario we're on. It's exponential increase in temperature. So it's kind of opposite that we're in Rome right now. I don't think anyone would have had any doubts back in the era of Italian fascism that there would needed to be a large-scale response. And right now, this is a struggle against money and power, simple as that. So the only thing necessary for the triumph of evil is for good men and women to do nothing. What can we do? We can look at it, we can grieve, we can talk about it everywhere all the time to people, to friends, to colleagues. We can teach and research about it. That's not so easy if you're a human brain mapper, but it is easy if you're a psychologist or social scientist. And we can advocate in our institutions, including in OHBM, in our universities, and including doing divestment from fossil fuels. All right, so with that, I'm going to switch to science. So the theory I want to put forward here in the little time I have is this idea that, and leveraging on quite a lot of things we've heard already, this overarching idea that decision and response conflict, stopping, surprise, switching, all these kinds of canonical functions of cognitive control all require a kind of pausing function. And that there may be a generic sort of computational pause function that is what's being done by these frontal subthalamic nucleus circuits. And here's a kind of overview, so quite simplistic and cartoony, but you've seen the basal ganglia diagram over on the left here. And the idea is that different kinds of functions such as stopping, conflict, and action selection are all involving somewhat different circuits between cortex and STN, but the underlying functional role is common. And this is inspired, of course, by the work by Haynes and Haber on there being some degree of functional topography. So I'm going to start out now by just talking about the stopping part a little bit to lay the ground. So many of you know this already, but the stopping action is something we do all the time. And in the laboratory, it's operationalized with the stop signal task. So in this case, what happens is that you get, for example, the letter T, and you just say T as fast as you can. Sometimes, as you're about to say the letter T, it turns red and you have to stop. So that's illustrated over there. And I can't use the mouse cursor. Okay. The idea, to summarize quite a lot of stuff, is there's some degree of consensus in the literature that there is an architecture underlying stopping, including the right inferior frontal gyrus, subthalamic nucleus of the basal ganglia, downstream effects on pallidum and thalamus, and that there's a hyperdirect pathway between right IFG and STN, and there's quite some evidence for that idea now, which I don't have time to get into. For example, stimulating STN and seeing responses in cortex within 3 milliseconds. And an overarching principle that I want to get across to you in a bit of data now is that it looks like the beta band, increased power in the beta band and beta bursts are an operating principle for this particular circuit. So how do we know that? Well, for example, we had back in the day recorded ECoG electrodes over right inferior frontal cortex in patients being monitored for epilepsy. And what you can see in this diagram here is that time 0 is when the stop signal occurs, so you have to stop the response. And you can see that there's an increase of beta band power around 16 hertz when you stop compared to baseline. That's not so much there on the fail stop trials, and that's a significant difference. Now, what's kind of intriguing or useful, actually, is we see something extremely similar in skull PEG. Obviously, with skull PEG, we're not going to make a claim about anatomy, but there's a right frontal component in all the studies we look at, 5 or 6 studies, where when we look in skull PEG, when people have to stop a response, you can see pretty much exactly the same pattern as an ECoG. So here you're seeing time 0 is the time of the stop signal, and there's that beta band power increase greater for success trials than failed trials in 2 different samples, about 15 to 20 hertz, and sometimes it's higher. We also know from LFP recordings from the basal ganglia in these patients that have an electrode in there that we see basically the same pattern there, which is that after the stop signal in the beta band, there is a relative power increase. And this is other work from Peter Brown's group showing the same thing. You start out by having a desynchronization in beta, but when you have to stop, it ticks up radically compared to the failed trials. So both IFG shows a relative beta increase, STN shows a relative beta increase. And now we've been doing some work, this is very preliminary, there's only a few patients so far, where the surgeon at UCLA, Paratian, is able to, on the operating table, put a strip over IFG and record the LFP from the STN at the same time. And there's a power increase in IFG, there's a power increase in the STN. And what's extremely intriguing, in just 2 patients right now where we have coverage of both, is that this unsuccessful trials when people stop, there is significant coherence between IFG and STN in the beta band, not in other ranges, not other times. So I don't know how all of this works exactly, but it seems like there's an intriguing emerging picture here about the significance of the beta band, maybe communication in the beta band in this particular circuit that's specific to stopping. Now, an enduring idea, I don't know if this is true, but lots of people have this idea, particularly in neurology, is that the STN has a very broad impact on the pallidum. The evidence base for that is pretty weak as far as I can understand, but it's this kind of thing. Old-style tracing, inject a bolus of tracer into the STN, and what you find is a relatively broad innervation of the GPI. That's kind of some of the empirical basis for this broad effect. So whether there really is an anatomical basis for the broad effect or not, I don't know, but we certainly have some functional evidence for a broad effect when you have to stop. We know this because we place the TMS coil over M1, give a pulse, and you measure the motor evoked potential from the hand, as many of you know. This is a measure of corticospinal excitability. Basically, you're measuring the excitability of the hand representation in the brain. Now, if you do this during the stop signal task, you get someone to see the letter T, and they're going to speak T as fast as they can. Sometimes the letter T turns red, pulse comes, and they're doing this, of course, in the vocal domain, so they're stopping speech, but you're measuring the excitability of the hand, which is irrelevant to speech. And when you do that, what you find is that that motor evoked potential at the hand is significantly reduced on trials where they stopped speech compared to other type trials. And that's the data over on the right, but this works for when you stop hand, you measure from the leg. When you stop eyes, you measure from the hand. In every study we've seen, we get the same thing. There is a broad scaleto-motor pause across the entire system whenever you stop an action in this basic paradigm. So we can ask the question, can we link this global motor suppression to the STN? And the way we approached this was to basically repeat the same study where people initiate a movement, say K, and then if it turns red, they have to stop, just like I showed you. We do TMS over M1, measure the motor evoked potential, but this was done in patients with Parkinson's disease where simultaneously we're recording the LFP from the STN. So I already showed you what those data look like. There's a beta power increase in the STN when you stop, as other studies have seen. Also, we find in the hand, when people successfully stop speech, we get exactly the same pattern of data as in healthy young individuals. So the really interesting question is, can we link these? So what we did here was we went to the period after the thing turns red, stop. There's a little bit of time to grab the beta oscillation that's there, and we can ask the question, on trials where there's more global motor suppression, what's going on with the beta band oscillation? And what we find is that those trials where there's more beta power in STN are those in which the global motor suppression effect is stronger. So this certainly links these things, but it doesn't prove that the STN is implementing this global motor suppression, but that's my working hypothesis. Now, I'm going to shift gears slightly from this to move to a related case. When we stop in the real world, we don't usually have stop signals. No one says stop, or usually not. You do that to small children, I suppose. But mostly, I spend a lot of time doing that, actually. You're supposed to encourage them and not stop them, but anyway. So you're standing on the edge of the street. You're about to step into the street in Rome. It's a dangerous business. And suddenly there's a bus there. That's what stopping looks like in the real world. Or you're about to say something, unflattering perhaps about someone, not that we would ever do that, and suddenly there they are. right? So stopping in the real world doesn't exactly involve stop signals, it involves unexpected events. And so one of the things we've discovered, or to some degree, is that this whole system is triggered not just by stop signals in the stop signal paradigm, but by unexpected events, and there are many kinds of unexpected events. I don't have time to get into that. So the way this is operationalized task-wise is you get ready, and you're going to say K or T, except every time it's preceded by a sound. So usually it's a sine wave for 200 milliseconds, so you get doot, K, K. Very occasionally we switch up that sound, for example, for a little bird song. Okay, what happens is there's post-novel slowing, people respond more slowly. But what's intriguing is we see the same right frontal beta power increase very quickly in the case when there's a novel sound compared to a standard, the same signature as when they stopped action outright. Now, an interesting question is the STN part of the circuitry causal to this way in which an unexpected event produces slowing or the behavioral interrupt or the pause? So this is work we did with mouse optogenetics. I'm just going to give you some highlight news here. This is the STN glutamatergic projection neurons are infected by channel rhodopsin. You can shine the light, and what the mice are doing is they're engaging in bouts of licking. So they're very hungry. They just randomly start licking. The bouts usually like 5 or 6 licks long. And after the second lick, the laser comes on. And what that does is it interrupts the animal. It stops them, if you like. But much more compelling than that is when we flip the logic around and we use halorhodopsin. So now the STN projection neurons, the glutamatergic projection neurons, are infected by halorhodopsin. That means if you shine the light on, you're going to silence them. This is like a functional lesion of the STN with temporal specificity. The paradigm now is the mice are licking, and after the second lick, we surprise them, okay, which basically produces an interrupt. The key manipulation is that on some trials, after the second lick, the laser comes on, silences the STN, then there's the unexpected event, okay, and what happens is then they're no longer interrupted. So you can see the data over here. If there's no surprise and no laser, then the percent of 3-lick bouts is very few because the animals lick 5, 6, 7 times. If the surprising stimulus occurs, then they have more short bouts because they get interrupted. If the laser comes on and silences the STN and then you give them a surprising event, you see the green bar is lower. They do not get interrupted. So we took this as causal evidence that the STN implements a kind of pause that produces interruption of behavior. There's a wider story here that I don't have time to get into, which is the idea that this kind of pause is not only affecting behavior, but it's also interrupting your cognition. So we may have evolved in a scenario going through the African savannah, all kinds of ideas in mind about getting food to eat and water from the river, and suddenly, little rustling in the leaves, behaviorally relevant, not a startling response, but a behaviorally relevant response. You're going to stop in your tracks and you're going to dump your mental contents. That would make sense. So the theory is that a lot of cognition is being maintained by reverberating thalamocortical drive. You see over on the left over there. And then an unexpected event occurs, in this case the deer. And then this very circuit I've been describing, involving IFC, STN, hyperdirect pathway, kicks in. Except the suppression that occurs is not simply motoric, it's so broad on basal ganglia output as to actually erode the mental content. So this is a theory of inducing distraction. And this is for another day, but we have operationalized this in the mice. We can have mice holding information across a working memory delay. We can turn on STN, channel rhodopsin. We can use the halo method. And our preliminary evidence are showing indeed that the STN are causal and, I suppose, necessary in some degree sufficient for actually interrupting cognition. So in the last little part here, I just want to switch briefly to conflict and say, I've talked about stopping, but we can go to conflict, and we've heard about that earlier. And here the prediction is that this is probably a somewhat different part of the STN, but it's a similar kind of circuit. Again, we can record from now pre-SMA and STN in the surgical setting, and they're doing a flanker paradigm where they have to respond to the central arrow, which is pointing right, but at the same time they've got left flankers, and they show very good behavior. And what we find is that they're in the pre-supplementary motor area this time. There's increased power when there's conflict. In the subthalamic nucleus, there's increased power when there's conflict. The pre-SMA is 504 milliseconds, the STN 688. But this is not the beta band. This is low-frequency oscillations of 3 to 6 hertz. So the takeaway from this is that conflict also recruits the system to pause motor output to buy time to make a decision, as we've heard earlier. But it seems to be a different circuit, and it's got a different oscillatory pattern, even though it may be a similar kind of function. The final thing is that we've done a TMS study with a Simon task, which induces conflict between right and left tendencies. The TMS coil is placed over the head, just like I showed you. Motor evoked pulse from onto the hand, and they're doing the task with the feet, so the hand is irrelevant. And what we find, which is pretty cool, is that at 250 milliseconds, after a conflict is detected, there is a global motor suppression of the entire system. But if they fail to correct it, then there isn't. So it looks as if this pause that comes on that has this very broad effect is functional to resolving errors. So the conclusion is that I've shown you that stopping and decision conflict involve related circuits but have different beta versus low frequency oscillation signatures. Another prediction would be that the dorsal SDN, which is connected up with primary motor cortex, also is a hyperdirect pathway, also plays a pause function, probably in the context of action selection. Even though there's uncertainty about the topography of SDN, and there's a lot more complexity as we've seen, I think if we could get enough subjects, enough patients, and enough resolution about where the contacts are in these different territories, we might get dissociations across all of these domains. So I guess just to summarize, I've gone a bit long here, I'd sort of read this. Whereas cognitive neuroscience has proceeded with different camps studying different tasks and different functions such as stop, switch, surprise, working memory, attention, everyone's got their thing, these results suggest that this frontal basal ganglia pause function is, I think, a kind of core primitive that's cutting across all of these sorts of domains, and I think that's one of the exciting things in cognitive neuroscience, that we should be focusing on the core primitives that cut across tasks rather than studying a particular task or function. There are many open questions about this, but I would guess that recruitment of the system is occurring every minute of the day in multiple ways, including as I'm speaking right now, and variations in the timing, the triggering, and the intensity with which the system is recruited is probably going to explain a lot of individual variability, whether it's related to stuttering or gait or more psychiatric things. Thank you.